steps in rDNA technology. Okay, these are the various steps in rDNA technology. Finally, how we are producing a transgenic organism, or how we are producing microbes which have a foreign gene in them, or genetically modified organism. So how we are producing it and all the steps are neither. First we will see the steps and then we will study each step one by one in detail. So what are the various steps? The first thing is isolation of the desired DNA. So when we have to introduce a foreign gene to an organism, first we should know which gene has to be introduced and from which organism we can get that gene. So getting that gene, a desired gene from an organism, that is called as isolation of the desired gene. Then fragmentation. Upon number GV and the DNA, the entire DNA is not needed. We need only a part which is actually needed. So the other part should be removed and we need only the part that is the desired gene. So for that we use the method called as fragmentation, which is done using restriction enzyme. We now know what is restriction enzyme. Next step is separation. Now we have a mixture of fragments. നമ്മുടെ ജീൻ ആവശ്യമുള്ള desired gene ഉണ്ട്, അല്ലാതെ കുറെ fragments ഉണ്ട്, but we need only the desired gene. So for separating that we use the method called as gel electrophoresis. Then നമുക്ക് ആവശ്യമുള്ള desired gene നമ്മൾ gel electrophoresis വഴി separate ചെയ്തു. Now we have to amplify it. നമുക്ക് കിട്ടിയ ആ ഒരു ജീനിന്റെ ഒരുപാട് കോപ്പി നമ്മൾ ഉണ്ടാക്കി വെക്കണം. Valuable ആയിട്ടുള്ള സംഭവമാണ്. We should not lose it. So we make many copies of it by the method, that is our process നമ്മൾ amplification എന്ന് പറയും. That method is called as PCR technique or polymerase chain reaction technique. Then, so we have made enough copies of the foreign gene or the desired gene. Now we are going to join it to the vector. Or directly we are going to give it to the host organism. So that is done using the enzyme DNA ligase to join. That is called as ligation. Now we are transferring it to the host organism either directly, that is direct method, or through a vector. Then we call it as an indirect method. Then we have to culture. Culturing is done when the host organism is a microbe. Okay. Another plants, so plants lab tissue culture methods are used. Another microbes we use huge vessels called as bioreactors, in which the microbes can be cultured or grown, and then after that we will do extraction. The desired products, that is the recombinant proteins, are extracted from the microorganism from this bioreactors. Then we do what is called as downstream processing, which involves a set of steps that we will study later. So these are the various steps involved in the rDNA technology.